Okay. Now, what is the second one? Chrysantic glomerulonephritis. So, chrysantic glomerulonephritis, also known as RPGN, what is rapidly progressive glomerulonephritis. So, again, here we can found this nephritis. So, nephritis means inflammation, okay, or what we say chrysantic glomerulonephritis. So here is our inflammation. So what we will find, okay, in chrysantic there is some, before we draw this diagram we have to know this terminology. So what is this chrysantic? Chrysantic is half moon like structure. So this is our moon, okay, and this structure is known as chrysantic, okay, half moon like structure. Now first point you got. So this is our parietal epithelium, okay. So when I say chrysantic, so we have to know some points. What points? First point is this glomerulonephritis will be segmental. So let's write here, this will be segmental. Segmental. So what do you mean by segmental? Okay, so in a mini diagram, if you see, this is our glomerular tract, and let's suppose this is our parietal epithelial cells, and in between we know the Bowman space. If now let's suppose this is our all capillaries, okay. If the pathology is in only one part of this glomerular tract, so think about this is affected or injured, so like maybe this is injured, okay, and this part is normal. So this kind of scenario is known as segmental, okay. Now chrysantic, what will happen here in chrysantic? So again, if there is any inflammation, there should be some infiltration and proliferation, okay, you got it. So infiltration of what? So you can see a little bit of infiltration, but not a major. So this is not a major portion in infiltration, but proliferation is here major, okay. So this is how you have to define. In particular scenario, you will see some particular case or histological presentations, okay. So this is the first basic point you have to know before drawing the diagram. Now we will draw first our crescent. So this is our crescent. So in this crescent, so I hope you can find this crescent. So this is our crescent, okay, half moon like structure, okay. This is our crescent. Now when we say this chrysantic glomerulonephritis, so translate it like this, ki half moon like structure has nephritis. So affected part or the injured part will be the half moon like structure. It means the opposite portion will be normal. Yeah, opposite portion will be normal. So now think about, this is our capillaries, And in these capillaries, you already know we will find some mesengium and mesangial matrix. Yes, mesengium and mesangial matrix. So this part is normal. The crysantic part will be abnormal. So in the crysantic part, what we will find? We will find this proliferation, okay? This proliferation. So first we have to know what will proliferate. The proliferation will be our epithelial cells. So epithelial cells will be proliferating here. So now there is a lots of cell here. Okay, and this is what you will find in histological slides. So it's very easy to like recognize, like you can easily identify this crescent and you can easily say, okay, this is our RPGN or Chrysantic Glomerulus. Now let's move to the next one. Next one is membranous nephropathy. So we will move to membranous nephropathy. Okay, so in membranous nephropathy, what we will find? First, you have to know again some important basic points. So first is in early stage. Early stage, it will appear normal. Okay, early stage, it will appear normal. In the later stage, if you talk about the later stage, it may be a thick glomerular capillary wall. Okay, it will be a thick glomerular capillary wall. But what we have to understand, how do we evaluate this histological slide. Again, let's go with basic again. So if this is our parietal epithelium, and now we got our normal glomerular capillaries, and we have little bit of mesengium everywhere lingering. Okay, now the main point we have to understand here is it is normal. Okay, the structure will be normal, or the capillary wall is thickened. So there is any infiltration, any proliferation? No. So like if the capillary wall is only thickened, like how we are going to define it? So we use here a special stain, and the special stain is known as silver stain. Okay, so silver methanamine stain or silver stain. So how you are going to define it? So the histological slide will be normal, and around this capillary wall you will find a black stain. Okay, so again, normal histological slide, and this around this capillary lumen, if you find this black stain, what is it? Membranous nephropathy. Membranous nephropathy. So you have to remember just some points here, how to evaluate these things. Okay, so it's very easy. Okay, normal and black, you can say ring around capillary lumen, and that's all, normal. So here you can see this Bowman space is also normal, right? you can easily find this Bowman space also. So it's normal, just the stain, the silver stain, and it will be black around the capillary. Okay, now then the another point is, fifth is minimal changes disease. Minimal changes disease also same, kind of same, not same. We got her minimal changes disease. So in minimal changes disease, as you can see, minimal, kind of minimum, or there is no changes. Actually there is no changes in light microscopy. So in light microscopy, basically you cannot find any changes, or actually it is normal. So again, if you see this capillary tuft, and if you see here this mesengium, and this is what it looks normal, perfectly normal, until or unless you go to electron microscopy. Okay, otherwise it is perfectly normal. Now we will go to FSGS, focal segmental glomerulosclerosis. Good one, this is good one.